Alright, so the body has a way in which it stores excess glucose. So glycogen is a major source of storage. So glycogen is for storage. Okay. Okay. So it's a storage form of carbohydrate in animals. All right. So glycogen actually resembles starch in a way. However, glycogen is a source of storage that the body uses. And where is it stored? So it's stored in the liver as well as the muscles. All right. Then, however, we also have exceptions like the brain, the heart, the kidneys, the adipose tissues, and even the red blood cells. So for these, they store very small amounts. So we have the RBCs, we have the brain cells, the adipocytes, which are the fat cells. So these equally store glycogen, though it's in very small amounts. Okay, so because glycogen is of very great mass, because it's a long chain, okay, you can picture this as a very long chain of tightly packed glucose molecules. Because of its great mass, the muscle contains about 3 or 4 times that much of glycogen as the liver. All right, so if this is your muscle, right, this is your skeletal muscle to be specific, it contains about 4 times that much of glycogen that can be stored by the liver. All right, so the muscle glycogen is a source of glucose during glycolysis, like we had discussed during muscle contraction. So whenever the muscles are contracting, um, the body uses up that glycogen that was stored in the muscles in order to provide it with energy. All right, so glycogen is present in the cytosol granules, okay, as well as, like we had said, it's found in the liver. So the liver glycogen stores increase during the well-fed state. This is to say, whenever someone is from eating, the levels of the glycogen stores increase because there is a limit. For instance, if the body can only store about 1 gram per mole of glucose, immediately this target is reached, the rest is stored as glycogen. Okay, so this is about glycogen. So the next thing that we're just going to talk about is why store glucose as glycogen? Why not just excrete some and use up the rest, or store and so on and so forth? So there are 3 reasons as to why we store glycogen. Why we store glucose as glycogen. All right. So just going to get into talking about those reasons. So reason number 1 is because fatty acids cannot be released from fats as rapidly as glucose can be released from glycogen. So glycogen is an excellent source of energy. Okay, in as much as it is excellent, it's also quick to be oxidized, or rather to be broken down in order to give us energy. The second point is that fats can not be used to store as a source of energy in the absence of oxygen. So when you look at fats, they cannot give us energy if at all oxygen is absent. Okay, however, for glycogen, there is no need of oxygen to give energy. Okay, so this means these glycogen stores can provide you with energy at any time, whether whether oxygen is present or not. So you may be asking, why are we referring to fats. Why are we comparing glycogen to fats? It's because fats are a secondary source of energy of the body. Whenever the body is in need of energy, we look to fats as well when the glycogen stores are depleted. Okay, so this is to tell you that the glycogen is the first source of energy that we go to. So fats cannot be converted to glucose, all right, which is required by the brain and the erythrocytes. Fats can't be converted to glucose. All right, and then when you look at the brain and the red blood cells, for them they strictly need glucose, otherwise they have a very difficult time actually functioning in the absence of glucose. That's why glycogen is very important. Okay, so we are just going to go straight into um, talking about glycogenesis. Okay, so we're going to talk about glycogenesis at this point. Oh, sorry. So this is glycogenesis. Okay, so under glycogenesis, we're going to talk about several steps that are involved in order for us to form glycogen. So this is a scenario where someone has eaten and now they have excess glucose, and some of that excess glucose has to be uh, converted to glycogen. So firstly, I'll start by talking about glycolysis. So glycolysis, the end product is it gives us ATP. Alright, and the first thing that we start with is glucose 6-phosphate. Okay, glucose 6-phosphate is the first thing that we start with. Now, whenever glycolysis occurs and there is excess ATP within the body, it means the body no longer needs to make more and more ATP. So what happens is that the reactant, which is the glucose 6-phosphate, it goes into what we call the glycogenesis in order to store that glucose to a form that cannot be easily used up at any time unless when there's a need for energy. All right, so the first thing is that glucose is phosphorylated to what we call glucose 6-phosphate. So I'll just put as glucose 6-phosphate, and this is catalyzed by the enzyme hexokinase. So hexokinase is the first enzyme that's phosphorylating glucose to glucose 6-phosphate. So this hexokinase is the enzyme that phosphorylates the glucose in the muscles. But when we're talking about the liver, we have what we call glucokinase. So glucokinase is the one that phosphorylates. That's when we're talking about the liver. Alright, so the next step, when we now have this glucose 6-phosphate, remember that it cannot leave because it has been phosphorylated, it cannot leave the cell. Alright, so the next step is that the glucose 6-phosphate undergoes isomerization. So it's isomerized, meaning there's a change in the molecular structure. So instead of glucose 6-phosphate, the phosphate is on carbon number 6. In this case, it's changed and it goes to carbon number 1. So we call it glucose 1-phosphate. Okay? And the one that isomerizes is phosphoglucomutase. So this is phosphoglucomutase. You can tell even from the end of the name, mutation, there is isomerization that's taking place. Okay, so from glucose 1 phosphate. The next step is that the glucose 1 phosphate reacts with UTP, which is uridine triphosphate. This UTP gives us what we call UDP glucose. Okay, and this UDP glucose, it's a glucose donor. All right, it's a direct glucose donor, and it's this process of it reacting with this is being catalyzed by this enzyme that we're calling UDP glucose pyrophosphorylase. Phosphorylase. Okay, sorry about that. So a pre-existing so when we have this now, when we have the uridine diphosphate glucose, which is a direct glucose donor, what you're going to have is the UDP glucose. Okay, now in order for us to have glycogen, we're supposed to have another glycogen molecule which is supposed to react, which is supposed to be added on. So there is what we call priming. You just add on to the already existing chain. So that already existing chain is called glycogenin. So we have what we call glycogenin. So this glycogenin is a pre-existing glycogen molecule. Okay, so this is actually a very short glycogen molecule. Now remember that this molecule that we had talked about is a glucose donor that we had talked about. So this glycogenin has the ability to add on glucose to itself. All right, so yeah, we call it autoglycosylation, meaning that that molecule can add on glucose to itself. So it transfers the glucose from this UDP that we talked about to itself. So this one donates the glucose. So the glycogenin keeps on growing, and when it reaches about 7 to 10 glucose residues, when it reaches 7 to 10 residues, the glycogen synthase is the one that takes over. Alright, so the next thing is that, so let me just write it here. You have your glycogenin, right? So the next enzyme that takes over is glucose synthase. Sorry, glycogen synthase. So this glycogen synthase takes over. All right. So it takes over in the adding on of these glucose molecules to the already existing glycogen chain. So it catalyzes that reaction of alpha 1,4 glycosidic bond. All right. So one glycosidic bond between the carbon 1 of the activated. So this bond is between carbon 1 of the activated glucose. So let me just write of the activated glucose, this UDP glucose, all right, and the carbon 4. So this bond is between alpha 1,4. So the glucose on carbon number 1 as well as on carbon number 4, there is a bond that's being formed there by glycogen synthase. All right. So whenever this is happening, a UDP molecule is released. So glucose is always added to the non-reducing side. Why is it non-reducing? Because it cannot be easily tampered with. So we add on glucose to the non-reducing side of that chain, of the chain. of the glycogen chain. All right, so whenever we've now, so let me just draw something like this. So this is your chain, right? This is your chain of glucose molecules, for example. Now this chain has grown to an extent, so the body finds means and ways of actually storing this glycogen in the body, because it has to be compacted in order for it to fit into the cells of the liver as well as the muscle. So there is what we call the 1,4 alpha glucon alpha glucan branching enzyme. So this branching enzyme forms the alpha 1,6. So between carbon number 1 and carbon number 6, it adds on. So if this is branching, you find that it will branch off here. Okay, let's do this. So it branches off somewhere here and adds glucose molecules. For instance, this is just an example. So if it's branching off, the branch is between carbon number 1 of this chain has been added to carbon number 6 on the chain below. So the bond is called alpha 1,6 glycosidic bond. Now this happens at regular intervals, meaning after it reaches a number of glucose molecules, it forms these branches. Alright, so the branches grow further. So it's like a tree where you have several branches going forth there, there, and there. It's not growing in height, but it's growing in branches so that it may fit within the cell. That's the whole purpose of the branching enzymes. So the branches grow further and further, added to the 1,4 glycosyl units for further branching so that it can be compacted. So the glycogen has branches connect on average 10 to 14 glycosyl residues, sorry. So if this is your chain of the glycogen, they have to be 10 to 14 in order for another branch to start. So when this branch starts, it reaches 10 to 14. At that end, another branch starts, just like that. Okay, sorry. So the branch goes there 10 to 14. Another branch is going to come over here, just like that. That's how this happens. So because of it being highly branched, or like a tree-like structure, it's this is what causes it to be soluble. All right, for instance, if you see a tree with a lot of branches, you can easily cut it by starting with the branches. Okay, that's the whole point. The best example I can give is actually of this very tree. If you have a short tree, right, and this tree is highly branched, it goes to many extents. All right, so just a minute. Okay. So if this is our tree, now this tree is very highly branched, so its branches are reaching as far as we can count, and these are the roots within the ground. What you notice is that this tree, for someone to easily pluck fruits, if it's a mango tree, they can easily pluck fruits because the branches are very close, they're very nearby. Okay, so this means even in the body, glycogen can easily be accessed because it's branched. Those glucose molecules are branched, they can easily be cut off. Unlike if the tree was going to be very tall, it would take you time just to climb all the way there and get a fruit. That's the principle that we are actually using right now. All right, so this is it. So the branching keeps on increasing and increasing depending on the amount of glucose that has actually been taken. All right, so from this we're now going to talk about glycogen degradation. So if glycogen can be synthesized, it means it can also be broken down. So we have what we call glycogen degradation. Okay, so this is our glycogen degradation. So automatically from the title, you can tell that now we are trying to cut off those branches in order to give us energy. So this is a case where someone hasn't had food for days and the body is trying to break down that glycogen that was stored in the body to give them energy. So we also call it glycogenolysis. Okay, glycogenolysis. So glycogenolysis takes place in the cytosol. So this is our cell. This is what we call as the cytosol. This is where glycogenolysis takes place. It takes place in the cytosol as well as in the lysosomes. So lysosomes are organelles that are found within the cytosol. So there are special enzymes that we're going to talk about in this glycogenolysis. The first one is glycogen phosphorylase. So we have glycogen phosphorylase. So this glycogen phosphorylase, it catalyzes the rate limiting step. Whenever you hear rate limiting step, this is where there is regulation of a system. You can either turn it off or you can turn it off. You can turn it on or you can turn it off. So it catalyzes the rate limiting step in the glycogenolysis by promoting the cleavage of these glycogen residues, the alpha 1,4 bonds. Okay, so remember we talked about the alpha 1,4 bonds. So what this glycogenolysis does, glycogen phosphorylase does, is that it catalyzes the breaking down of these bonds. Okay, and if these bonds are broken, it means we can now have glucose 1 phosphate. Okay, so we can have we're able to yield glucose 1 phosphate. So the terminal glucose residues, these glucose residues that are at the far end, are sequentially removed, meaning they're removed one after the other until we remain with about 4 glucose residues. 1, 2, 3, 4. So if this is a very long chain, we cut them until all the glucose molecules are reduced to a point where we only have 4 glucose residues that will remain at the side of the branching. So remember we said it's a long chain. Now we have 4 glucose molecules that are close to where the chain is now branching out. Okay, we'll have something like this where we were branch. There's a branch point here. This is our branch point, our alpha 1,6 glycosidic bond. So the hydrolysis of this linkage that you're able to see here requires what we call the debranching enzyme. So this debranching enzyme will cut off the linkage of alpha 1,6. Alright, now the debranching enzyme has a name. We call it amylo. Sorry, let me just write it down nicely. Okay, so this is amylo alpha 1,6 glucosidase. Okay, so this is a debranching enzyme. It releases one glucose molecule. So that it will release one glucose molecule here, and to further phosphorylase action can proceed. So the whole point is once it breaks this bond, remember we talked about the glycogen phosphorylase. So once it breaks that bond, the glycogen phosphorylase will now continue doing its job by breaking these bonds now. Okay, it will keep breaking those bonds one by one until the glucose residues are released, until we reach with about 4 alpha 1,4 glucose bonds attached to a a branching point, just like we had. Okay, so the combined action of this glycogen phosphorylase, it combines with what we call the glucan transferase. Okay, the glucan transferase transfers a triglyceride unit from one branch to another. So it gets a triglyceride, meaning 3. It will get this. Remember, this was broken, right? This part has been broken. So it will get 3 from here. Then get the 3. It will get these bonds. These 3. Let me see if I can move. So it will get them one by one. One by one, bring them here. Okay, sorry though, this one is not perfect, but it does something like this. So it gets these 3, takes them there. Okay, gets this bond. So it gets the triacylchloride unit from one branch to the other. So it gets it here and combines with the other one. Alright, so whenever it does this, it exposes this point that had a branch right there. So whenever these two before we even go further, let me just clarify on the debranching enzyme and what they were just simply trying to talk about. Okay, so there is an enzyme that we call glucan transferase. So what glucan transferase does is that it gets 3. Remember, we had 4 residues remaining, right? So it will get these 3. Remember, there's a branching point here. Okay, so what glucan transferase does is that it will get these 3 glucose residues that are attached to each other, then it will shift, bring them on top here. Okay, it will move them and combine them to this chain. Okay, so let's see what we are talking about. So those 3 will now be combined to this chain on top. Okay, we've now combined the 3. So the whole essence is that when this happens, this branching point is going to be exposed. Okay, this is the alpha 1,6 glycosidic bond. It remains vulnerable, and when it's vulnerable, this is when the debranching enzyme now comes to cut off this branch. Okay, I hope that made sense. Okay, so the combined action of the glycogen phosphorylase and the glucan transferase and the debranching enzyme is what leads to the complete breakdown of this glycogen. This is what breaks it down completely, because they are working together in order to release glucose. So the glucose 6 phosphate, remember we had glucose 1 phosphate, right? That was released. This glucose 1 phosphate can be converted back to the original form, which was the glucose 6 phosphate. Okay, now there's an enzyme which catalyzes this action. Let's use a different color. So this enzyme is called phosphoglucomutase. So when you see this, remember this is the enzyme that was even catalyzing phosphoglucomutase. So this is to tell you that this reaction is reversible. You can go back and forth. So this glucose 1 phosphate is converted back to glucose 6 phosphate. So in the liver, but not in the muscle, the glucose phosphatase. So there's another one that we call glucose glucose phosphatase. Okay, the glucose phosphatase hydrolyzes the glucose 6 phosphate. So this enzyme will hydrolyze. To hydrolyze is to use water to break a bond. So this glucose phosphatase um, breaks down this glucose 6 phosphate, and this happens in the liver, not the muscle. This is happening in the liver. Alright, it breaks it down, or rather it hydrolyzes the glucose 6 phosphate to glucose, and then that glucose can now be transported to the blood. This is your blood vessel. We have red blood cells over there, so it can now go into the blood system and transported to several other places to give, or rather to generate ATP. Alright, so all this is happening in the cytosol. The next one that we're going to talk about is lysosomal degradation of glycogen. Okay, degradation. Alright, okay, so we have what we call lysosomal degradation of glycogen. So under this, we won't talk so much about it, but we'll say something. So there's a small amount of 1 to 3% of the glycogen. There's an amount, about 1 to 3% of the glycogen is continuously degraded by the lysosomal enzyme. So the lysosomal enzyme is called alpha 1,4 glucosidase. Alright, so these are lysosomes. You know that they contain within themselves several enzymatic or other catalytic enzymes within them that are able to just break down several things in the cell. So only a small amount go into the lysosomes for degradation. Alright, the next thing that we're going to talk about is regulation. Okay, so there's what we call regulation of glycogenesis and glycogenolysis. So just write as regulation of glycogen metabolism. Okay, so this is to tell you that glucose is not constantly being produced. Okay, there has to be a limit be produced, and when can it not be produced. So you've learned about several hormones like glucagon, insulin, epinephrine. We're going to talk about all those hormones and how they work. So the first one that we're going to talk about is cyclic AMP. So just write it as CAMP. So cyclic AMP is a very important factor. It integrates the regulation of glycogenolysis, which is the breaking down, as well as the glycogenesis. So it can regulate as to whether glycogen should be formed or whether it's going to be broken down. So the principal enzymes controlling glycogen metabolism are glycogen phosphorylase. Remember, these were the rate limiting. So we have glycogen phosphorylase, and we also have glycogen synthase. Okay, so glycogen synthase, remember we had talked about it in glycogenesis, right? But for glycogen phosphorylase, this one was talked about in glycogenolysis, okay, which is the breakdown. That's why our regulation is going to be centered on those enzymes, because how they function is going to determine um, as to whether glycogen will be broken down or it's going to be built up. All right, so these two enzymes are the ones that are regulated. So there's what we call reversible phosphorylation. So what we mean is that whenever an enzyme is phosphorylated or dysphosphorylated, will determine whether it's going to be active or not. So these things work opposite each other, okay? And we're going to talk more in details. So these enzymes are actually either phosphorylated or dysphosphorylated in response to the hormones that I talked about, the glucagon as well as the epinephrine, and so on and so forth. So cyclic AMP, okay, this camp is actually derived from ATP, all right, by adenyl. So adenyl cyclase, all right, and it acts in response to the hormones, the epinephrine and the glucagon. This is how camp comes in the scenario. So whenever we have increased camp levels, so we have camp over here, this arrow is representing there's an increase of camp levels in the body. It leads to the phosphorylation of proteins. So this one will cause phosphorylation, meaning there'll be adding on of a phosphate group. So phosphorylation of proteins, okay, in the activation or inhibition. So they'll either be active or inactive depending on what type of protein that we're talking about. Remember, enzymes are proteins, so those are the ones that we are referring to. So we refer to glycogen synthase. So when we talk about glycogen synthase, it exists in its phosphorylated. So we're going to do this. When I write a phosphate, whenever glycogen synthase is phosphorylated, it becomes inactive, okay? But whenever, so this is positive, it's phosphorylated. But when it's not phosphorylated, it becomes active. So in here it's inactive. Sorry, when glycogen synthase is phosphorylated, it's inactive, hence the arrow. Okay, so these are quite opposite. When you have a phosphate, the enzyme is inactive. When you don't have a phosphate, the enzyme is active. Okay, so phosphorylation of glycogen phosphorylase. So let's write the other enzyme as well. Okay, so we have glycogen phosphorylase. Okay, whenever this one is phosphorylated, let's put a positive. Whenever it's phosphorylated, this one acts in the opposite. It's going to be active. And whenever it's not phosphorylated, it becomes inactive. So this one is, we can say it's directly proportional because they're following each other. If there's a phosphate group, it's active. If there's no phosphate group, it's inactive. But for glycogen synthase, that's the direct opposite of what's happening. So with having understood what these enzymes do and what conditions them being active or not, we'll talk about insulin. Okay, so insulin promotes glycogenesis. Okay, so if insulin is promoting glycogenesis, right? So this one is promoting glycogenesis. How is it going to do that? For this to promote glycogenesis, what does it do? It's going to activate the enzyme. So insulin activates glycogen synthase, because if glycogen synthase is active, it means we have more of glycogen being produced. All right. So insulin will promote glycogenesis by activating the glycogen synthase. Okay, how does it do this? It does that through the action of a protein that we call phosphatase. Okay, so insulin is working hand in hand with a protein that we call phosphatase. So this is phosphatase 1 to be specific. Number 2 point, we're now going to talk about glycogen phosphorylase. Okay, since we've written it, let's just talk about it. So glycogen phosphorylase is also inhibited by glucose 6 phosphate. This is glucose 6 phosphate, right? I'm sure you're wondering how come the reactant is now inhibiting glycogen phosphorylase. So glucose 6 as well as ATP, which is a high energy signal in the cell, these they inhibit glucose 6 phosphate. Okay, so these are the inhibitors. Next up, we are just going to talk about the what happens in the liver, in the muscle as well. So start with the what happens in the liver is that glucagon leads to the formation of cyclic AMP. So this is glucagon, right? Glucagon will lead to the formation of cyclic AMP. Now cyclic AMP activates the phosphorylase. And you know what phosphorylase? Phosphorylase breaks down glycogen. Okay, this is how they're connected. Glucagon will be released by the liver, and then it will produce cyclic AMP. Cyclic AMP is going to activate the phosphorylase, and phosphorylase will cause the breakdown of the glycogen, thereby giving us glucose in the body. So all this is happening in the liver. Let's now talk about what's happening in the muscles. Okay, so what's happening in the muscles is that the muscle lacks both enzymes, like, so there is no glucagon phosphatase. So muscle glycogenolysis is activated in response to epinephrine through the beta receptors. So it uses beta receptors instead of those other receptors. So epinephrine is the one that works in the muscle. So this is your muscle cell, right? There are several receptors that are receiving signals. Now these signals only respond to epinephrine because of the beta receptors that are found on the muscles. Alright? So usually this happens whenever when someone is scared, let's say they're being chased by a dog or they're in danger. The body has to act fast. So they act in a fight situation or prolonged exercise, because now the muscles have to start using the glycogen that they stored up. And having said this, we also have other mechanisms that are hormone independent, meaning they don't function. Okay, so these are hormone
So we talk about glycogen storage disease.
Okay, so under glycogen storage disease, we talk about several things that lead to these diseases. So there's a series of inborn errors of metabolism that are involved in the glycogen storage. So these diseases are affecting how glycogen is supposed to be stored. So they result from when those enzymes that are involved to store glycogen are affected. Okay, those that are involved in glycogen metabolism are affected. So muscle glycogen phosphorylase. So let me write it down. So this glycogen phosphorylase, it differs genetically. All right, it's genetically different from the glycogen phosphorylase. So if this is for the muscle, right, and this is for the liver, of course, same enzyme, but they differ genetically. So some degradation of glycogen also occurs within the lysosomes, like we had said, when the glycogen particles become surrounded by membranes that then fuse um, with the lysosomal membranes. Okay, so you have a case like this. These are the glycogen molecules that become surrounded with the membranes of the lysosomes. These are the ones that are going to be degraded by the lysosomes. All right. So the enzymatic defect in the Gierke disease. So let's look at this. We call this case von Gierke.
Muscle pains in the joints. This is the type 1. So there's under pump disease. There's a defect in lysosomal glycosidase. So lysosomal glycosidase. Okay, so this one is the one that's called the type 2.
This is a type 2 storage disease. So this one leads to the accumulation of glycogen particles in large membrane enclosed residue bonds. So you know that the membrane is like this, it's around an organelle. So the glycogen stores are going to be, they'll accumulate in the membrane of the lysosomes. Remember when we talked about the lysosomal degradation, all right? So the accumulation of this glycogen affects various body tissues, and most of them affected, we have the heart, the skeletal muscle, the liver, as well as the nervous tissue. All right. So the last one that we talk about is the Cori type. So this is the Cori type 3 disease, storage disease. So the structure of the liver and the muscle of the muscle glycogen is abnormal. Instead of it being branched and whatnot, it's abnormal, and it accumulates, impairing the function of the tissues. So here we're looking at the liver. Okay, the liver is accumulated with glycogen that's abnormal, and this is going to it's going to impair the function of the tissues that are found in the liver and the muscle. So patients that have this type of um, disease, the Cori type, they lack the debranching enzyme. So here it's involving the debranching enzyme. Okay, now remember the debranching enzyme is the one that breaks those branches. So you have these branches and then there. So if this enzyme is not there to break that bond, only the outermost branches of glycogen will be effectively utilized. Only these that are at the terminal end are going to be utilized, meaning these at the branch points are going to be held inside, they will not be used up because the debranching enzyme is missing, all right, or rather is defective. So only a small fraction of the abnormal glycogen is functionally active and accessible to store the glucose, all right. So another one that we'll talk about is McArdle disease. This one is type 5 glycogen storage disease. Okay, so this is type 5 glycogen storage disease. So what happens is that under this, it experiences no other symptoms but unusual fatigue. So the person begins
Glycogen is the first reservoir for the support of blood glucose
levels. So a deficiency, or when the glycogen phosphorylase is missing, in other type of disease that we talk about, we call it the HARS or type 6. This is type 6. Okay, VI. So this is type 6 storage disease. This one results in fasting hypoglycemia. Fasting hypoglycemia. Okay, so hypoglycemia is usually mild because patients can still synthesize glucose from um, glycogenesis. That's why it's mild. So accumulation of glycogen leads to liver enlargement. So we also have liver enlargement here. Okay, due to accumulation of glycogen. So this is the end of glycogen metabolism. Hope you enjoyed the lesson.